happy mapping. It is a technique to map the genome where the position of two or more genes it can be mapped on the genome or chromosome. So in more technical sense, here we can analyze the haploid genome of an organism, and during analysis the fragments can be amplified by using polymerase chain reaction. So that's because of this reason the haploid genome is analyzed. This technique it got the name of happy mapping. In other words, we can say that haploid genome means here that the low concentration of the target DNA it has been analyzed after fragmentation. Because low concentration or minute quantity has been analyzed after series of dilution, so we have expected that we have analyzed the haploid genome, or in other words low quantity of DNA. So that's why from haploid the name to this technique it is given happy mapping. Now this technique it have removed several limitations that were present in RH mapping or radiation hybrid mapping. RH mapping have several limitations. One of the limitation is that here the generation of few cells or hybrid cells, it is difficult. Then some distortions they may be there in different parts of the DNA or DNA sequences or centromeres, and then it is difficult to distinguish between the genomic DNA that belongs to the irradiated cells and the host cell. So all these limitations they arise as a result of in vivo technique, because here we are going to use the living cells in which one has been irradiated. So in vivo system is there, so such distortions or limitations they may be there. Whereas on the other hand, happy mapping is a bit modified RH mapping technique, and here we are not going to use the hybrid cells for the analysis of genomic DNA fragments. Now how we can proceed for happy mapping? The DNA fragments or the genomic DNA that we want to map, it must be isolated, and here it is prevented from fragmentation during the process of isolation. मिसाल के तौर पर हम सेल्स को डायरेक्टली लोड करते हैं एग्रोस जेल पे, और फिर हम सेल्स को ट्रीट करते हैं with certain chemical reagents like proteases or detergents that can cause the lysis of the cell when the cells they are embedded in the gel, so that the fragile DNA that will be liberated, released after lysis, it will not be fragmented. So during lysis, the protein debris or other molecular debris that will be produced, they will be diffused away by the gel, and the DNA molecules that are much larger, they will be trapped within the gel. Now when the gel contains the target genomic DNA, then it can be isolated from the gel and then exposed to fragmentation by using different techniques. One of the method is that mechanical shearing it can be used to cause the fragmentation of the DNA fragments. Another technique is that it can be exposed to gamma radiations that can cause its fragmentation. So the length of the fragments or the number of fragments that will be produced, it is dependent upon the dose of such radiations or mechanical forces. Now we have DNA that is now fragmented, then it can be diluted into different concentrations, so that different concentrations can be loaded in different microtubes, that 100 samples may be diluted and it will represent a panel for that mapping, a particular panel that is going to be used for mapping. Because here the DNA concentration has been lowered by several steps of dilution, so minute quantity of DNA has been analyzed, and this minute quantity may represent only one DNA landmarks or DNA sequence. So because of this lower concentration of DNA, it has been suggested that it may represent the haploid genome, और इस वजह से इस टेक्निक का नाम, जैसे कि मैंने पहले बताया, हैप्पी मैपिंग जो है वो इसको दिया गया है. Now we have DNA fragments. Then by using some specific STS sequences, it can be amplified and it can be separated on agarose gel, and different landmarks, they can be separated. So this is also a good variation of RH mapping to map the DNA, and it has removed all the limitations that I have mentioned that were present in RH mapping.